Okay. Thank you so much. So I am Judie Magura. I'm a postdoc at AHRI. I think I was one of the very first users for 10x Genomics when it came to South Africa. I would like to thank the organizers as well for giving me this opportunity to share how I've been using the 10x platform for my work. And in today's talk, I'll just introduce you to what I do and why I do it. I'm going to highlight the 10x Genomics platforms that I use and just show you a little bit of the questions we've been able to answer using 10x. And the title of my talk is Mapping B-cell Precursors in HIV, a Southern African Perspective on Germline Targeting Vaccine Strategies. Okay. All right, just trying to move to the next slide. Okay. So to start off, I just want to answer this question. Okay, we have a little bit of a lag, but we are getting through there. Okay. So the first question that I would like to answer is what is the germline targeting approach? In a nutshell, this is an HIV vaccine approach to induce antibodies against HIV that are broad, meaning antibodies that can neutralize many strains of HIV. And then why is this important, or why does it matter? Why do HIV vaccines matter anyway? I think the short answer is because we still don't have an HIV cure. We know that AHRI has been very good. It has been a success, but we still have more than 1.3 million new infections each year. We have made really good progress with the long-term injectables, and these have managed to do away with the burden of having to take pills every day, but they're still very costly and they might be very impractical to implement, which brings us where we are, where we believe that an HIV vaccine will remain the most cost-effective solution to HIV. Okay. So what does the germline targeting approach mean? What is it, right? In principle, the idea is to use engineered immunogens or vaccine candidates that are engineered in the lab, or to just form vaccine candidates against HIV/AIDS, right? So with this HIV vaccine candidate, what we want to do is to design an immunogen that is able to activate or wake up a specific kind of B cell. Then after that, we bring in a series of other immunogens, engineered immunogens that have got increasing complexity. And using those immunogens, we want to guide the starting B cell into producing mature, long-lived, broadly neutralizing antibodies, right? So from this perspective, we can see that the starting B cell is very important, or this strategy actually depends on the starting B cell. So with that, we come to the aim of the study, right? So what we are trying to do is to say, in the pool of B cells that we have, or in our starting B cells, do we have the B cells that have got the right kind of features? Do we have B cells that have got the potential to become broadly neutralizing antibodies or not? That is what we are looking for. And for this study, we are not only looking at the pool, but we're looking for a class-specific B cell. Class-specific because we know that all broadly neutralizing antibodies have got defined features, and they have been characterized already, and we know what they look like. So we want to be able to say, from this class-specific B cell that we have, does it match those features or not? What is really happening, right? The other question that we want to be able to answer is to say, does the HIV disease have an impact on this B cell repertoire? We also want to be able to answer the question of, does the timing of AHRI initiation, when we introduce AHRI, does it also have an impact on this B cell repertoire or not? Right. So the samples that we are using to answer these questions, they come from the fresh cohort. This cohort is established in Durban, and it's a cohort of female participants that are aged between 18 and 23 that are at high risk of HIV acquisition. These participants are followed for a very long time, and they're tested for HIV twice every week, right? So because it is a longitudinal study, it means we have samples from a time when they are HIV negative, and then at further times when they become people living with HIV, right? So for our study, we are looking at PBMCs that are starting at HIV negative. Then some of these participants become HIV positive and they're put on treatment early. For some of them, they're put on treatment late. Actually, this changed after 2016. I think the guidelines to ART initiation had changed in South Africa, so it didn't have to be this late. When we then converted to the treat as soon as the individual is detected or HIV is detected in the participant. There's two graphs here that I'm showing on top here. I just wanted you to see the viral load as well as the CD4 count kinetics in the two treatment groups. So in the early treated group, we have ART initiated immediately within 5 days of infection, whereas in the chronic treated group, we have ART initiated maybe a year or 2 years later. Okay, so for this study, the first step, of course, for us is to isolate the B cell because we are looking for a specific kind of a B cell, a specific type of a B cell. So our first step is just to isolate that B cell. Okay, so we start off from the PBMCs from our participants, and then we use a magnet selection. We are trying to enrich for just B cells because we are particularly just looking at B cells which generate broadly neutralizing antibodies. So from those enriched B cells, what we go on to do is then to stain with the appropriate antibodies for B cell selection using flow cytometry. We also include total-seq. So total-seq is hashing antibodies. These antibodies allow us to multiplex our sample. So we're able to process up to 5 or more donors at a time. But because we want to be able to sort out specific B cell, we also include the EODGT8, which is an engineered vaccine candidate. So this is all included in our staining panel. Once we have stained our cells, the next step for us is then to do the flow cytometry so that we can isolate our B cells of interest. What I'm showing here is just our gating scheme. So we start off with the viable B cells, and from there, we just want to be able to isolate the EODGT positive B cells, meaning B cells that bind to our vaccine candidates. We also want them to be negative for the knockout. So the knockout is the CD4 binding site, which is exposed in our candidate of interest, is marked here. So there would be negative for the knockout, but positive for the candidate of interest. The one thing that I want to highlight here is that this kind of cells are very rare. So I'll give you an example. For a typical sort, we usually start off with about 14 million of the viable B cells, but we usually only end up with about 245 cells. So these are very rare, but because we're able to multiplex, it means we still start off with the minimum required amount of cells for 10x. After we have isolated our B cell, the next step for us is to then to generate the 10x library. So this is where I am using 10x Genomics. I am using it to do my single cell RNA analysis. Right. So I start off with the pooled B cells. I subject that to the 10x Chromium so that I can generate the gems. Then from there, I go on to create the libraries, my single cell RNA libraries. I'm particularly interested in the VDJ library. But what we appreciate from the 10x Genomics is because we are able to do sample hashing, it means I can add as many samples as possible. I can add as many donors as possible. I don't lose out on the fact that I'm only sorting for a specific class of B cells that are very rare. The other thing that I also appreciate for the 10x Genomics is that I am able to come up with the paired antibodies, right? If I was using bulk sequencing, for example, I would end up with only the heavy chains on their own and the light chains on their own that are not paired. But by using 10x Genomics, I get the paired antibodies, which allows me to be able to define the specificity of the antibody. It allows me to be able to characterize a definite class of the antibody, which is what I'm looking for. Okay, once I've generated my RNA libraries, I then go on to do the sequencing. At the moment, we are using Illumina platform for sequencing, right? The things that I really like about 10x Genomics is that post-sequencing from my raw data, they have very, very, very good post-analysis support. So from my raw data, I am able to put this into Cell Ranger, clean my data, do the QC, and just figure out what's going on. So for me, I use the VDJ loop browser because I am interested in the specificity of my antibody. So I want to be able to know which V gene is it using, which D gene does it use. I'm also interested in the CDR length as well as the sequence of that CDR. So loop browser really allows me to pinpoint what it is exactly that I need from the data that I have, and it is useful for me, right? Then from there, now we have usable data. So here I'm just going to highlight some of the things that we've been able to do, trying to answer our question whether we have the correct bNAb precursor in our pool, also trying to answer the question whether HIV disease has an impact on these precursors and whether the timing of AHRI has an impact on the precursors, right? So the first thing that we looked at was from the EODGT positive B cells from our flow cytometry, do they use the correct VH gene usage, right? As I mentioned before, for every bNAb class, it is defined. So we already know the features that it uses. We know the specific VH gene that it uses for a class. We also know additional features. So for the VRC01 class that we were interested in, why were we interested in this? It's already in clinical trials. It has already been proven to work. So we wanted to know whether this also, the precursors to this protein neutralizing antibody were present in our cohort. So the first thing was just to look at the VH gene usage, and we could see that we do have the gene of interest. We also have other genes that have been defined for other HIV bNAbs, so that was good. Then from there, we went on to look at the light chain. So we wanted to check whether this heavy chain pairs with the correct light chain that has got a 5 amino acid. We only identified those in the early treated group. So the other two groups, which is the HIV negative and the late treated group, they do harbor the correct heavy chain and the correct light chain. The only missing thing is that they do not have the correct length for that particular light chain, meaning that they do not fall in this class of interest, right? Then from there, from the early treated group, we went on to just investigate further whether it meets all the other correct criteria or the defined features for the bNAb of interest. So we could see that we had this group being rare. We only had about 5.92% of the VH-2 gene, which uses the STAR02 anneal. From that percentage, right, the ones that also harbor the correct light chain, we only had about 9.5% of those. So they are still present. They are there, even if it's a lower percent. But we expected this because we know that this class is very rare. The next thing that we then went on to do is to look for this signature sequence logo in the light chain. So for every class of a mature VRC01 bNAb, it has the signature logo, which is the QQYEF. So this is what we were particularly looking for in our early treated, whether we have this signature logo, so that we would have the correct light chain, the correct heavy chain, as well as this signature motif. So yes, we did have the correct signature motif in our early treated group. And so just to give you an idea of what does this mean in terms of vaccine strategy, right? This is what we think is happening from the results that we got so far, what we are predicting. Because we didn't see this signature 5-amino acid in the HIV negative group, as well as the late treated group, we think that in the HIV negative group, we do have the correct features. We do have the correct precursors. They're just not fully there, right? But because we see the signature logo and we tick all the boxes for the VRC01 class in the early treated, we think that that disease progression into early treated, it sort of drives the precursors into some form of maturity. And because these individuals are put on ART treatment early, that those features are preserved in that group. We are predicting that we don't see them anymore in the late treated group in the chronic phase of disease, because probably they are now depleted, or maybe that disease progression ends up depleting those features that we are interested in, right? So to conclude what I've been talking about, we wanted to see whether we have what are the germline targeting vaccine implications, right, in our cohort. We know that we have the correct gene usage in across all groups. We only see the signature, although for the VRC01 class in the early treated group, this is lacking in the HIV negative and late treated. So we think that early treatment may preserve the VRC01 class potential. We also think that vaccine targeting for the CD4 binding site could be feasible in this cohort because we have the correct features. They just need to be guided or be driven in the correct direction. For further directions, we are hoping to do functional validation on the antibodies that we are isolating from those B cells. With that, I would like to acknowledge our panelists and thank the participants, the fresh participants, particularly that have helped us to do the study. The individuals from the TUMBI group, as well as all our collaborators across Africa and US. Thank you.